All right, let's continue on chapter 4. We ended up when we started talking about chapter 4 about just DNA structure. So, in terms of double-stranded DNA, right? The reason why the adenine and the thymines and the guanines and cytosines are always together is that when we look at the actual structure of DNA, we want some uniformity. So, we wouldn't want anything that's really bulky on one side and then another real bulky base on this side. So, you can imagine if you have adenine on this side and then on this other side, it's guanine. Well, now you have two very bulky bases facing each other. Not only that, then if adenine is with guanine, that means that thymine is with cytosine. So you'll have two bulky bases together facing each other, and then you'd have two of the small bases facing each other, which will make it so then there's no uniformity at all. There'll be regions of the DNA that will be much larger and expand, and other areas of DNA strands that are going to be thinner, right? And it's just going to keep bulging and then thin, bulge and thin, right? So there's no uniformity when that happens. So it makes sense that, you're not going to have adenine with guanine, right? You're going to have adenine then with thymine or cytosine, large base with a smaller one on each side, right, facing it. Now, why we have adenine with thymine, guanine is with cytosine, that particular configuration is due to the fact that we have the ability to form maximal hydrogen bonding. When we have adenine, thymine, guanine, and cytosine, we maximize the number of hydrogen bonds. And remember, it is just these hydrogen bonds that stabilizes the actual, right, the actual strands so that they don't separate. And if the strands of your chromosome separate, that's it, it's over. You cannot reform them. So we need these hydrogen bonds to be, first of all, multiple, to be strong, to be facing each other, and to be maximized. So then we can have the most amount of stability possible here. Now, in terms of DNA structure, we kind of mentioned it before, 98% is non-coding. That means it doesn't code for any one protein. And remember what we mentioned, what a gene is, right? So just because it's non-coding does not mean that it is not important. Because remember, just as important as how to make the protein, which is what a gene is. A gene tells us which of the 20 amino acids goes into each of the different locations required to make a proper protein. So with 20 amino acids, we need to know which of the 20 goes into location number one, number two, number three, number four, number five, all the way to number 50. Once we get that right sequence of those amino acids in place, now we have the primary structure of the protein. And once you make that, everything kind of falls in place. But you need to make that accurately. So what we need to do then is have a gene that kind of tells us how to make it, but that only accounts for 2% of our total DNA. The remaining 98% is doesn't code for anything, but it's just as important because it tells us when to make the protein, how much to make it, and if possible, have a duration of how, just how much and when, right? So we kind of mentioned how when you have insulin, you want to make a lot of it in a very short amount of time. And importantly, what happens if you don't make enough in that short amount of time? Well, you have some insulin resistance and you're starting to have type 2 diabetes. What happens if you make too much of it for a prolonged period of time? Then you might get hypoglycemia and we might have you go into a diabetic ketoacidosis, or you might even go into a comatose state. So we need to be very careful of when we make it, how much to make it, so then we can have not just areas that are very important for just the actual coding for genes, but to regulate that gene. So 98% of the DNA is for that. Now, when we look at the difference between double-stranded DNA, we wrap it around the proteins that we then produce chromosomes. Now, the chromosomes, when you see ones that have two arms, each arm is called a chromatid. We call them sister chromatids. The weird thing is, once we physically separate the two arms, they are now different chromosomes again. The problem is, once they're kind of stuck together, they are different chromosomes. All right, so we kind of went through this chromatins right here. Histones are the proteins which you are wrapping around the double-stranded DNA, so then you can make sure that that long, long, long spool of thread that is your double-stranded DNA. Remember, double-stranded DNA, each one of them represents a chromosomes. So we have 46 double strands of DNA. And each one of them, if we allow it to just unroll, is about 4 to 6 inches. So when you're talking about something that's 4 to 6 inches, and now you somehow have to pack it into a nucleus that is small on top of it, not only that, but the micros, the cells we can't even see with a mic unless we have a microscope. So the nucleus is incredibly small. So how do we package everything in there? We have to roll it tight on those histones. Once we roll everything tight in the histones, and we have the double-stranded DNA that now we can store in the nucleus. Perfect. That's why the histones are so important. Nucleosomes zigzag like an accordion, chromatin thrown into complex irregular loops and coils, 1,000 times shorter than the original double-stranded DNA. So by doing all this, you really compacted 1,000 times what it was previously. Right. Chromatin and chromosomes. Each chromosome is packed into its own region of the nucleus. In a non-dividing cell, the chromatin is not static. That means it's not just sitting still. There changes moment to moment according to genetic activity of the cell. What do we mean? When you have a cell that's maybe like living in your pancreas, right? The chromatins, the chromosomes there are not just sitting still. You'll have protein synthesis that occurs at specific chromosomes at specific times. So you'll have certain genes that get turned on. Again, examples with insulin production. So when you're eating, right, the gene is going to be turned on. So the chromosomes will be unrolled at that specific gene, and then we'll have protein synthesis and all the enzymes required for that to happen, activated only for that specific gene. Remember, you want to produce a lot of that protein, insulin. So what we need to do is turn on the gene, package as many, RNA polymerase and make as many messenger RNA as possible so then we can produce a lot of proteins. But it takes a lot of energy. So there's no point turning on other genes when you don't need to. So if you don't need that protein, what's the point of making it at that point? So you'd just be wasting energy. So only the genes that are needed will be activated so that we can produce the proteins that are needed at that moment. But the chromosomes or chromatins themselves, they're not really just going to sit still. Each chromosome consists of two parallel filaments of identical DNA. We call them sister chromatids, and this happens right before we have cell division. So when we have sister chromatids, that means that we have already undergone DNA replication, we've doubled the DNA, and now we have what should be two chromosomes. But since the chromosomes are linked together by the centromere, we call them sister chromatids. Chromatids are joined by a constricted centromere. The centromere is a protein, and we're going to have something called kinetochores, protein plaques on each side of the centromere, and the centromeres are what guides the proteins. So then are what guides the chromosomes. So then we can locate it centrally. So then when we divide the chromosomes, each cell gets one copy of one chromosome from each parent. So we'll talk about that coming up right here, right? So this is, when we look at this right here, this is our double-stranded DNA, a copy of that same double-stranded DNA. This one chromosome, here's another one. When they're linked by the centromere right here with a kinetochore, right? When they're linked like this, right, what we're going to see is that because they're linked, held in place, we call each one sister chromatids. We don't call them sister chromosomes. Now, once we split it in half, even though it might be a micrometer, right, in distance, or even a nanometer in distance, just as long as we see a little break here, each arm counts as separate chromosomes. But when they're linked together, it only counts as one chromosome, and we consider each leg of this a sister chromatid. Now, RNA structure and functions. Besides DNA, we have RNA. RNA are much smaller, and it resembles DNA in that it's got basically the same bases: adenine, guanine, cytosine. The only one that gets replaced is thymine. There's no thymine in RNA. Instead, there's uracil, right? Now, the thing about RNA is usually they're functional, right? So in terms of DNA, there's only one type. It codes or it regulates a coding for the proteins of when and how to make it. So, and how much to make it. RNA is the actual activation of it. When we want to make a protein, it is these RNA molecules that will be formed that will be used to make the actual protein we need. There are three important RNAs for protein synthesis. There's messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA is going to be important because it makes photocopies of the DNA gene. Then we can make multiple messenger RNA per gene. Remember, when you're eating and you have this huge amount of glucose going into your bloodstream, we're talking about hundreds and hundreds of trillions of individual glucose molecules. It's going to go into your bloodstream. Now we have to get it into every cell of your body, and you have trillions of cells in the body. Why am I mentioning this? Well, we only have a very small selective number of cells in your pancreas that produces insulin. All right. Because we have such a small number of cells that produces insulin, how do we make enough of it? If each cell only has one gene that codes for insulin. Well, what we're going to do is in a process that we call transcription, we're going to take the gene and we're going to read it and we're going to make complementary base pairs from it. And in the process, we're going to make lots and lots of messenger RNA. There are two reasons why this is important. Number one, all right, by going and making photocopies, by reading the DNA gene, by making, by, right, by, let me just go back here, by reading the DNA gene for insulin, I have an enzyme that base pairs. So if I have adenine on one side, I'll have uracil on the other. If I have cytosine on one side, on the DNA gene, I'll have guanine on the other. If I have thymine on one side in my gene, then my messenger RNA that I'm reading is going to have adenine. So all I'm going to do is just base pair everything. In the process, I'm going to produce something called messenger RNA. So messenger RNA is a photocopy specific for the gene I'm producing at that time. I don't care about any other genes on that chromosomes. I'm not going to make photocopies of it because I don't need that protein. I only need to make the insulin one right now. So what's the point of wasting energy and producing even another protein that is not going to help me? So what happens? I'm going to put all my energy into producing insulin. I'm going to have enzymes, many of these enzymes we call RNA polymerase enzymes. And those enzymes are going to read the gene specific for insulin. As soon as one ribosome, as soon as one of the RNA polymerases binds, it's going to read and go down and base pair. Then the second one is going to bind to that same gene and read and base pair, and then a third one. Well, what happens is like when you have multiple RNA polymerases all reading the gene at the same time, you're going to make lots and lots of photocopies of messenger RNA. So instead of just one copy of a gene and everybody's trying to read from that one copy, well, what's going to happen is I'm going to make photocopies of it, and not just one photocopy of the gene. I'm going to make millions of them at a time, right? And each cell is going to make hundreds and hundreds of millions of photocopies of that gene. That photocopy is messenger RNA. I'm going to transfer that messenger RNA out of the nucleus into the actual cytoplasm where I have ribosomes right there, or ribosomal RNA that can bind to it. And the same ribosome that we saw in chapter 3 that's important for protein synthesis, these ribosomal RNA then is going to bind to the messenger RNA. It's going to form active sites. It's going to give you the structure required so then we can bring the proper amino acids in place. Right. So the ribosomal RNA, they're the ones that provide the structure so then we can bring the proper amino acid for the bases that code for it. Now, the transfer RNA, that's the RNA that actually brings the proper amino acids in place. We know for all proteins, every single one, in the first location of amino acid, it always starts with a amino acid that is called methionine. So when we read that messenger RNA is kind of a start codon, and the start codon is a combination of three RNA bases. Is adenine, uracil, guanine, AUG, right? AUG is our start codon. And once we have AUG, the messenger RNA has that as a starting point. Ribosomal RNA binds to that photocopy. Then transfer RNA brings the methionine in place that matches AUG. Perfect. Now we have a perfect match. Now, in terms of protein synthesis, I want you to know when do we have, there's two, know that, all right, when just in generalities, right? When a gene is activated, messenger RNA is made. A messenger RNA, like we mentioned, is a photocopy of the gene. It's complementary to the gene, right? Now, it's formed in the nucleus, then it's going to migrate to the cytoplasm where we're going to put everything together. In transcription, DNA codes for messenger RNA and occurs in the nucleus. So in transcription, number one, it occurs in the nucleus. Number two, we're going to take the DNA gene and we're going to make lots of photocopies that are complementary RNA base pairs of it, right? Not just one. From that one gene, I'm going to make millions and millions of messenger RNA. Then I'm going to take all those messenger RNA out of the nucleus because you can't make the protein in the nucleus. And once I take it out of the nucleus, then I'm going to put it on rRNA, ribosomal RNA, which are the ribosomes. And the ribosomal RNA is going to start reading the messenger RNA. And it's going to bring the proper amino acid to the correct positions by bringing the proper transfer RNA to the proper positions. And that happens in the cytoplasm, right?